Okay. Good afternoon, everyone. First, I want to thank the workshop organizing committee to invite you join this discussion, this workshop, which has been an exciting learning opportunity for me so far. And I'm an outlier here. I don't do any ontology work, but I definitely is someone who can potentially benefit from all the exciting new tools that our three panel speakers just showed us. So today, what I want to do is to bring you an important problem, TB vaccine development, a long-existing global health need and a challenge. And hopefully you will be interested. Okay. So, and the reason I say TB vaccine development is a long-existing global health challenge and global health need is that, as reported in an article from Lancet in 1997, evidence of TB was found in the spines of mummies dating back to 3,400 to 2,400 BC. More recent phylogenetics studies suggest that Mycobacterial tuberculosis, the causative agent, was present in humans at least 15,000 years, had present in humans for at least 15,000 years. But despite the long history of TB as a human disease, TB remains a major global public health problem today. And showing by this global incidence distribution map from WHO's 2021 global TB report, and TB is currently present in every country of the world, and also regional disparity in TB burden is evident, given that 86% of new TB cases were in the 30 high burden countries, and 8 countries accounted for had 2/3 of the global TB burden. Furthermore, despite TB is preventable and curable, it is a leading infectious killer of the world's population. According to WHO, a total of 1.5 million people died from TB in 2020. It's of this 1.5 million, 1/4 is HIV infected. And it's until COVID pandemic, TB was the #1 infectious killer, even above HIV. So for an important global health issue, a leading infectious disease, definitely we need a vaccine to fight the epidemics. As we all know, vaccine represents the most cost-effective tools that has contributed to the reduction of the global burden of vaccine preventable disease. But unfortunately, TB epidemic control has not been benefit sufficiently from the vaccine that we have for TB. Believe it or not, the current only available TB vaccine was developed 100 years ago. BCG, some of you probably have got the BCG vaccine if you are from a developing country. And this is a vaccine made with a live attenuated strain derived from M. bovis in early 19th. So it's named after two scientists who developed the strain in early 90s. The pioneer vaccination of BCG took place between 1921 and 1927, but it was not until 1950s widespread vaccination started. And currently it's an EPI vaccine, WHO recommended the EPI vaccine, which demonstrated the significant effectiveness. However, protection was not consistent across all forms in all age groups and all geographic areas. The only vaccine in use is the only vaccine in use for prevention of TB currently, despite it was developed a century ago. So, but another interesting thing about the TB vaccine is it has some demonstrated effectiveness in preventing other infection, other intracellular pathogens such as leprosy and other lung TB mycobacterial infection. Recently, there have been a lot of research trying to answer the question where BCG vaccination has a protection against COVID-19. And based on ecological studies, observational epidemiological studies, but the more random controlled trials are needed to definitely answer this question. So in order to develop a universally effective vaccine against all forms of TB and in all the populations, it is important to understand the factors that may have contributed to BCG protection variation. For example, BCG is known to be very effective against the severe type of pediatric TB, such as TB meningitis. However, it showed very little protection against adult TB, pulmonary TB, which is the most contagious form of TB. So this is what's currently known or currently hypothesized factors contributed to BCG protection variation. Genetic difference in BCG strains. Several speakers today mentioned the study variants of certain pathogens. Genetic difference in BCG strains. So one variant could be vaccine strains, and then also could have some difference among MTB strains, pathogens. And then methodological difference among the clinical trials. So especially from what I understand, the ontology study, when you're pulling studies from data from different studies, one of the important things is to look at methodological difference among the clinical trials or studies, because that could contribute significantly to the outcome of the studies variation. Then genetic difference within and among host populations. This could have an impact on the population's ability to respond to vaccination for good or worse, for protection, efficacy, or adverse events. And the varying levels of malnutrition among host populations. And especially if we are studying some disease involving underdeveloped regions, populations, varying BCG vaccine against the primary infection in children and endogenous reactivation of persistent infection versus exogenous reinfection. So for the very same kind of disease, it could, due to different pathogenesis or epidemiological mechanism. In the case of TB, for example, an active disease could be the result of a new infection obtained from ongoing transmission. It could also be due to reactivation of persistent infection. So this differentiates the mechanism is also important. And the effect of environmental microbacteria on the host immune response to BCG. And I think the thinking of this behind all these factors actually could be applied to any infectious disease at least, because all these factors do have a role in the varied response to vaccine, which will influence our decision on vaccine design. So what are the current strategies for TB vaccine development? And the next couple of slides summarize that. The current strategy, basically, based on the pathogenesis of TB, we need 3 kinds of vaccine in terms of the target stage of MTB infection. And one is the pre-infection, another is post-infection. And then these 2 types listed here are the primary goals for TB vaccine development. One is to prevent infection and or primary disease. This is very much the same or similar to many childhood vaccine. Basically, it's used for naive population who have not been exposed or infected by the by TB organism. So another type is target on people who already exposed to TB or who already had a latent TB infection. Basically, we want to stop, we want to keep the infecting agents in the dormant, not causing clinical disease. This addresses a unique challenge to TB vaccine development, also address a unique characteristics of TB, because 1/4 population is estimated to be latently infected. So the third one is more towards enhance the chemotherapy. And the potential new types of TB vaccine for a long time, it's the subunit vaccine, live microbacterial vaccine, and inactivated microbacterial whole cell vaccine. But more recently, mRNA vaccine is also explored. And based on the experience with COVID-19 vaccine, this is definitely a very promising platform for future platform-based vaccine and the TB vaccine too. Here, I just want to share a couple of resources for those of you who are interested in the status of the TB vaccine development, and then also highlight a few challenges and opportunities, and the mechanism of MTB infection, latency, and reactivation. Definitely is one of the very important questions, challenges, opportunities, given 1/4 of the world population is estimated latently infected. We definitely need the second type of vaccine, target and post-infection. And then determination of protective immunity against MTB. And one of the major problem for so long we don't have a new TB vaccine is because we still try to learn what's the protective immunity. In fact, some disease, even some already, were proven useful vaccine, we still learning the protective immunity. Of course, there are several ways to learn about the protective immunity. And then identification of co-infection and comorbidity in TB. After this, the most important is the TB-HIV co-infection and the TB and the diabetes comorbidity, because both HIV and diabetes could impact host immunity. How that changes the immune response to mycobacterial infection represent another important area of research. And then high rates of exogenous reinfection in humans cured of primary TB. So if nature infection doesn't protect, how we can do better with vaccine? This is another important question. And the limited studies of the difference in immune response to MTB infection and vaccine infection among children, infants, children, and adults, because the BCG vaccine showed significant difference among these age groups. So important ethical issues, of course, when you come to vaccine development, withholding BCG vaccine in clinical trials, testing novel vaccine in HIV prevalent population, ensure access of the infectious vaccine in high burden, low resource countries. These are more the downstream research once the vaccine is in use. At last, I just want to share a reference of our most recent work that takes advantage of all the bioinformatics vaccine ontology work that my lab did in collaboration with Dr. Edison and Dr. Host, proud former student, and who has done a lot of contribution to the field. Thank you.
Wonderful, wonderful talk.